Welcome back with another microbiology lecture from Microbial Genetics, which is RNA or ribonucleic acid. In this video lecture, we will discuss RNA, different types of RNA and its structure. Let's start. RNA is a single-stranded, but sometimes in some viruses, double-stranded RNA can be found. The basis of RNA include cytosine, guanine, adenine, and uracil. In RNA, the uracil are present instead of thymine of DNA. In RNA, ribose sugar are present instead of deoxyribose sugar of DNA. And the nucleotides of RNA are joined by phosphodiester bond just like DNA. Now, types of RNA and their distribution in cell. Number one is messenger RNA, which are present 5%. Second is ribosomal RNA, that are present 80%. Third is transfer RNA, which are present 15%. Here are some other forms of RNA, which include hnRNA or heterogeneous nucleus RNA, which are the primary transcript of messenger RNA that contain exon and introns only. Fifth is snRNA or small nuclear RNA that participate in RNA processing. Next is telomerase RNA, which are involved in chromosomes replication. Last one is antisense RNA, which are involved in gene expressions. In cell, about 50% RNA is distributed on ribosomes and endoplasmic reticulum. 25% are distributed in cytoplasm, 15% are distributed in mitochondria, and 10% are distributed in nucleus. Before we starting RNA in detail, we must know about transcription and translation in both prokaryotes and in eukaryotes. Transcription is the synthesis of any kind of RNA from DNA with the help of RNA polymerase enzyme. They may be messenger RNA, ribosomal RNA, or transfer RNA, while translation is the synthesis of amino acids or protein from messenger RNA through ribosomes. Mostly in prokaryotes, transcription and translation are simultaneously, or we can say coupled transcription and translation, because prokaryotes have no nuclear membrane, while in eukaryotes transcription occurs in the nucleus and translation occur in the cytoplasm because the eukaryotes have nuclear membrane. Messenger RNA is single-stranded RNA molecules which are complementary to a gene on DNA. It contains a triplet nitrogen bases or 3 nitrogen base called codon, and each codon codes for a single amino acids. From DNA, a gene carry message for protein synthesis. That's why it is called messenger RNA. A single strand of RNA is synthesized in the nucleus of DNA template and then entered to the cytoplasm, where its genetic code specifies the amino acids sequence for protein synthesis. The prokaryote have coupled transcription and translation, while the eukaryote have separate transcription and translation, so both DNA strands have genes. A single strand of DNA is decoded by RNA polymerase enzyme, and messenger RNA is synthesized by a process called transcription. RNA is made by a strand of DNA called template strand or DNA template. Now, transfer RNA. For translation to occur, the transfer RNA transfer amino acid to ribosome and messenger RNA complex for protein synthesis. They are usually soluble, hence called snRNA. Transfer RNA have molecules about 70 to 95 nucleotides in length. When transcribed, transfer RNA molecule are present in a single large precursor, and the ribonuclease P enzyme convert the large precursor RNA into transfer RNA. The transfer RNA shows extensive base pairing and they are clover leaf-like structure. At 5' phosphorylated guanosine present. One important feature of transfer RNA is acceptor arm in which 7 base pairs are present. At the 3' end of all transfer RNA, CCA sequence are present. At OH group of adenine or adenylic acid that forming an ester bond with the carboxyl group of amino acid. Another feature of transfer RNA is anticodon loop, which are 5 base pair long, and it is present opposite to acceptor arm. Their nucleotide sequence are complementary or opposite to anticodon nucleotide of messenger RNA. Another feature of transfer RNA is dihydrouridine arm or DHU arm, which are 3 to 4 base pair long. It serves as the recognition site for the enzyme called amino acyl transfer RNA synthetase. Another feature of transfer RNA is pseudouridine arm, which are opposite to DHU arm and are about 5 base pair long. It have role in binding of transfer RNA to ribosome. It is generally denoted by a Greek alphabet PSI. Last one is ribosomal RNA. Ribosomal RNA are present on ribosomes, which is made of protein and RNA. A prokaryotic ribosome or bacterial ribosome are 70S and are smaller than eukaryotic ribosomes, where S is a Swedberg unit and used for sedimentation velocity. Their large subunit are 50S while small subunit are 30S. They contain different RNA and specific proteins. The large subunit contain 5S RNA, 23S RNA and 31 proteins, while small subunit have 16S RNA and 21 proteins. The eukaryotic ribosomes are 80S and are larger than prokaryotic ribosomes. They contain different RNAs and specific proteins which are later transported to the cytoplasm. The large subunit contain 28S RNA, 5.8S RNA and 5S RNA plus 49 proteins, while small subunit contain 18S RNA and 133 proteins. Thanks for watching. If you like the video, subscribe my YouTube channel for upcoming latest lectures.